So that erythropoietic signal would be one of several signals that regulates hepcidin. We already knew at that time that there were iron signals that regulated hepcidin via the bone morphogenetic protein receptor signaling pathway. There was inflammation that was controlling hepcidin via the STAT3, JAK-STAT pathway, and the inflammatory mediators, including IL-6. But added to that then was an erythropoietic signal that somehow regulated hepcidin. The observation that something regulated hepcidin was then developed in both experimental models and in humans. And so what was done at that time is this was the group of Ashby et al. In 2010. They took some human volunteers, injected them with erythropoietin, and before and after the injection, monitored multiple iron parameters, including hepcidin, transferrin saturation, ferritin, and soluble transferrin receptor 1. And what they reported is that less than 24 hours after the injection of erythropoietin, there was an abrupt and profound drop in hepcidin. And that drop in hepcidin was not preceded by a change in iron signal, just as Clem Finch had observed. There, in fact, was a small drop in the iron signal, and we'll return to that later. And that is a parallel circuit to the one that I will discuss today. But the change was rather small, and before hepcidin dropped, there was no significant change in any of the iron parameters.
Tom, can I stop you for just 1 minute?
Please.
Your screen is presenting with a couple of black rectangles, and I don't know what those are. I'm trying to figure out what that's coming from. Yep, those right there.
That one?
Yep.
That is the video, so I can shut that down. Let's see if that can go away. How is it now? Still there?
It's smaller.
Okay, let's see if I can just completely remove it. That could be, I can almost completely remove it. How about now?
Yeah, that's it. That's better. There's a little one at the top, but it doesn't interfere with reading the slide. So thank you very much. Let's go forward.
Yeah, that was the video. I don't know why it shows through, but that is the video.
Okay, thank you.
Okay, so the erythropoietin elicits a drop in hepcidin, and that drop in hepcidin takes place less than 24 hours after the rise in erythropoietin. And it is not preceded by a significant change in any of the iron parameters. Let's see. So I was lucky that at about that time when we were contemplating starting this project, Leon Kautz showed up in our lab as a postdoc, and he drove this project to completion. And was a very excellent experimentalist and thinker, and really should be credited with this discovery. What he did is he took some mice, bled them or gave them EPO, and observed the course of hepcidin after the bleeding or the administration of EPO. And here's a time course. In the blue is the effect of a 500 microliter phlebotomy. In the red, there is an effect of the administration of EPO, erythropoietin. And no matter which you do, between 4 and 9 hours after the stimulus, there is a profound drop in hepcidin, which bottoms out at about 12 to 15 hours and then begins to return towards normal. And so the initial level of hepcidin drops about something like 10-fold, 8 to 10-fold, as a result of this intervention. And so we decided that we would look for something that has the opposite time course, a signal in the bone marrow that would essentially be the inverse of the signal, and that would begin to appear between 4 and 9 hours. And so we were looking for this green kind of a response. And we did that by transcriptional profiling of the marrow, looking at RNAs that change during this time, and selecting those that encoded secreted proteins. And at this point, and this is an important factor in science, luck favored us because there was only one such transcript that encoded a secreted protein. And we named this transcript Erythroferrone because, as you will see later, it connects erythroid activity to iron metabolism. And it had previously been known by systematic names such as FEM132B, CTRP15, and another group described the same factor as myonectin. And I put it in quotation marks because there were two different myonectins, and this particular myonectin was the same protein essentially as erythroferrone or FEM132B. And this protein belonged to the C1q TNF-alpha superfamily. It's a 50 kilodalton glycoprotein. And importantly, it was highly expressed in erythropoietin-stimulated erythroblasts. So I'll tell you a little bit then about Leon's work on describing this new substance. And as our transcription of profiling revealed, if you take mouse bone marrow and follow the time course of this particular mRNA after the administration of a stimulus such as phlebotomy or EPO, phlebotomy blue, EPO red, you will see that indeed, as the RNA profiling showed, the transcript goes up something like 30, 40-fold within 4 to 9 hours after the stimulus. And importantly, the signal is coming out of the erythroid lineage, and it is the black bars show you unstimulated bone marrow. The red bars show you bone marrow that was stimulated by either at EPO administration or bleeding, in this case bleeding. And what you can see is that it is the basophilic, polychromic, and orthochromic erythroblasts that generate most of the signal, most of the RNA in the marrow, that most of the erythroferrone RNA, as you can see here. So we then recombinantly produced this protein, or made it or expressed it in a lentivirus, and gave it to mice, and showed that in fact the protein was able to suppress the hepcidin mRNA within a few hours. And either the protein or the administration of the Erythroferrone lentivirus would suppress hepcidin. And most importantly, if we cultured primary mouse or human hepatocytes and treated them with erythroferrone, we would also be able to suppress hepcidin production by these hepatocytes. So we argued then that erythroferrone acts directly on the liver to repress hepcidin transcription. There was a mouse that we could purchase in which this part of the lexicon collection of knockout mice, in which this gene was actually knocked out. And so we bred homozygote Erythroferrone knockouts, heterozygote Erythroferrone knockouts in blue, and compared them to their wild-type litter mates, and then bled them or gave them erythropoietin, and observed that, as before, the wild-type mice profoundly suppressed hepcidin within 12 to 15 hours. But the heterozygotes only did half as good a job of doing the suppression, and the homozygote knockouts only had this very minor suppression at 12 hours, and essentially lost most of their responsiveness to bleeding. And so the hepcidin remained essentially level. And importantly, if you take these mice and bleed them, the knockout mice take longer to recover from the effects of bleeding. So the wild-type mice are bled down to a hemoglobin that's 4 points below the baseline, and they largely recover, or substantially recover, by 9 days. The Erythroferrone knockout mice are delayed by 2 to 3 days in that recovery. So we postulated then that erythroferrone is a stress hormone that is helpful to mammals and maybe other vertebrates as well for recovery from a hemorrhagic insult. But interestingly enough, it is completely dispensable for chronic response to erythropoietic stimulation. And this was shown by Richard Coffey, who would take mice in our lab and administer EPO over 2 weeks, giving 3 doses a week, so 6 total doses, then waited 15 hours and analyzed the mice. And what he showed that there was no difference in hemoglobin, serum hepcidin, or liver iron concentration between erythroferrone knockout mice and wild-type mice in their ability to raise hemoglobin in response to erythropoietin stimulation. There was no difference in suppression of hepcidin, and there was no difference in the terminal liver iron concentration. So these mice, as they make hemoglobin, these mice lower serum hepcidin and deplete their liver iron, and the knockout mice and the wild-type mice do that equally well. So we then concluded that erythroferrone is a stress hormone that has something to do with the acute response to hemorrhage. Then 2 other groups, Juan Rajesh and Chayu Wang, described then the mechanism that is involved in the action of ERFI. So several groups, including ours, were looking for a receptor, but and Hal Drakesmith's group, in collaboration with Pfizer, actually worked up this problem in a different way, and they found that rather than a receptor for ERFI that would regulate hepcidin, ERFI unexpectedly directly bound to bone morphogenetic proteins. So if you look at the signaling that controls hepcidin, sinusoidal endothelial cells somehow sense iron or responds to another cell type that senses iron, and they proportionately to the liver iron load secrete bone morphogenetic proteins 2 and 6, probably as a heterodimer in part. And the heterodimer interacts with the BMP receptor complex to transcriptionally stimulate the production of hepcidin. So in proportion to the BMP26 signal, hepcidin is transcribed and secreted by hepatocytes. So in order for sinusoidal endothelial cells to control hepcidin production, these molecules have to cross this small space called the space of Dicey. And in the space of Dicey, erythroferrone intercepts BMP2 and 6, presumably, and prevents its interaction with the BMP receptor. And so this mechanism was then postulated, and there's good supportive data for this. This mechanism is the most likely mechanism. There may be others as well, but it's the most likely mechanism that controls or inhibits the transcription of hepcidin and the secretion of hepcidin. And the ALK3 subunit of the BMP receptor may be particularly important in this interaction and in this regulation. So which part of erythroferrone is actually involved in trapping bone morphogenetic proteins? So we, and in parallel, Hal Drakesmith's group also had divided the ERFI molecule into 2 parts, one that we call fragment 1, the other one fragment 2. Fragment 2 is the TNF-alpha-like globular domain. Fragment 1 is this extended region here that really doesn't have much impressive structure in it, and really we did not expect it to be active, but as it turns out, if you do a Biocore sensogram of the N-terminal half versus the C-terminal half of ERFI, the C-terminal part, the TNF-alpha-like globular head, does not interact with the BMP26. In this case, we anchored the BMP26 heterodimer on the chip. the C-terminus does not interact with the surface, but both the whole protein and the N-terminal extended part strongly interact with the BMP26 heterodimer, as well as with BMP2 and BMP6 individually, and probably with several other BMPs. So this then was evidence that there's direct interaction between erythroferrone and the bone morphogenetic proteins, and this may be the main regulatory pathway by which erythroferrone regulates the transcription of hepcidin. So then, to summarize this part of the talk, we then postulate that erythroferrone is a stress hormone that speeds up iron mobilization early after erythropoietic stimulus. It promotes rapid recovery from hemorrhagic anemia. As I mentioned, there are other mechanisms that contribute. One of them was proposed by Mirziov et al. As I showed in one of the slides, there's a slight decrease in transferrin saturation after EPO administration. This is due to erythropoietin inducing the expression of TFR1 on erythroblasts with about the same time course as the induction of ERFI. So ERFI is induced at about the same time that TFR1 is induced on erythroblasts. This rapidly depletes the preferred ligand of TFR1, which is diferric transferrin, which is a small minority of the total transferrin forms. And the depletion of diferric transferrin then downregulates hepcidin, even if it has only a small effect on total serum iron. So the selective depletion of diferric transferrin decreases the BMP signaling and helps downregulate iron helps downregulate hepcidin in response to erythropoietic stimulation. ERFI is dispensable for the response to prolonged erythropoietic stimulation, and ERFI acts by interfering with BMP binding to the hepcidin regulating BMP receptor, and specifically the ALK3 component. Okay, so now I come to the second half of this talk, which is the pathological roles of erythroferrone. So we had developed an assay for measuring human and mouse erythroferrone, and then used the assay to study populations of patients or mice with beta thalassemia. And in this particular image, I'm showing you data on human thalassemia, in which the thalassemic patients were divided into three groups. Those that did not receive transfusions at all, or very rarely, in blue. Those that received regular transfusions but were measured before their scheduled transfusion, and those that were measured after their scheduled transfusion, and here in gray are the normal controls. And if you look at these groups, the patients who are never transfused had the highest, by far, the highest levels of erythroferrone. But when they are started on transfusions, their erythroferrone levels go down, and specifically, they go down immediately or within a day or two after transfusions. So this, again, illustrates that the kind of erythroid factor that was proposed by Clem Finch that would be responsive to, as he had proposed, that would be responsive to transfusions and very high in thalassemic patients, that this erythroferrone behaves just like that factor that Clem Finch had predicted must exist. A transfusion-suppressed erythroid regulator. And in fact, if you plot erythroferrone versus hepcidin, you get this hyperbolic curve in which the higher the erythroferrone in these patients, the lower the hepcidin, and interventions that lower erythroferrone end up raising hepcidin. So there is a kind of an inverse relationship in human patients in beta thalassemia between erythroferrone and hepcidin. And then in the murine model of beta thalassemia, which is the TH3 plus model, these mice, of course, are somewhat anemic to a hemoglobin of about 10 to 11, the red bars here, compared to the wild types in black. Their serum EPO is increased, So the wild-type mice in black, the red bars indicated the profound increase in serum EPO in these mice. And erythroferrone is increased both in the bone marrow and in the spleen, and the increase is of the order of what? Tenfold over the baseline mRNA levels, both in the marrow and the spleen. So the model recapitulates the human situation. These mice make higher levels of erythroferrone. And when we developed the ELISA for murine erythroferrone, we could actually measure these levels. And compared to wild-type mice or ERFI knockout thal mice or ERFI knockouts themselves, we could detect erythroferrone in the circulation of these mice. And if we made a model of thalassemia in which erythroferrone was knocked out, we could actually reverse the hepcidin suppression. So here are wild-type mice, here are thalassemic mice, and here are thalassemic mice with ERFI knocked out. And we could partly, and this is an interesting point, we could only partly reverse their iron overload. So there must be other factors that contributed to iron overload in these mice. But we could significantly decrease their iron overload by knocking out ERFI, but we could completely restore hepcidin. So this model, the TH3+ model, has some limitations. In these mice, unlike in humans, the iron overload is modest to moderate, and it is self-limited. So these mice become iron overloaded when they're 6 weeks old or so, and then after that, hepcidin rises, and after the age of 6 weeks, there is only very slow accumulation of iron. Most importantly, in humans, we see a range of erythroferrone response, but these mice have a homogeneous erythroferrone response because they're genetically homogeneous. So, we could not use these mice to study the effect of erythroferrone, mostly because the erythroferrone levels were rather low, but also because we could not isolate the effect of anemia from the effect of ERFI very easily. So to circumvent this problem, Richard Coffey, a wonderful postdoc in our group, decided to develop a transgenic erythroid overexpressing model of, that would overexpress erythroferrone in the erythroid compartment. And to do that, we used a globin promoter enhancer-driven plasmid, and we made transgenic mice that would overexpress murine erythroferrone, which is flagged with the flag tag so that we could tell the difference between endogenous and exogenous erythroferrone. And we made mice, we made 3 lines of mice. In one line of mice, there is a single transgene copy, and that expresses relatively low levels of erythroferrone. In the high line, that expresses 3 copies and expresses high levels of erythroferrone. And then spontaneously from the high line, another line was generated that expresses intermediate levels, which we call the medium line. And it has only 1 copy of the transgene in the same location as the high line. So if we look at ERFI expression in these mice, we can see that these are males versus females. Wild types are in open symbols, transgenes are in these pink or red symbols. And both the males and the females in the low line overexpress erythroferrone, and as you progress from the low line to the medium line to the high line, the mRNA expression increases about 2-fold between from the lowest to the medium to the highest line. And if you look at ERFI protein, it has a more dramatic effect on ERFI protein. If we use the ELISA, we can see that the serum ERFI, the low line is of the order of the thalassemic mice, maybe a little lower, or and the intermediate line is about 10-fold higher, and the hyaline is another 10-fold higher. Now, if we look at serum iron in these mice, we can see that these mice actually have dysregulated iron homeostasis. So starting with the low line, there is a significant and progressively increasing concentration of iron in the serum, going from about 20-something to 30-something micromolar. If you look at liver iron concentration, again, there is a progressive increase in liver iron concentration from the low to the medium to the high line, reaching about 3 to 4-fold baseline. Now, something funny happened here, but the point of the slide before, where there is some glitch in the transmission, is that that serum hepcidin is in fact suppressed in the high line, and there's a hint of suppression of serum hepcidin even in the intermediate line, but the low line does not suppress hepcidin. But if we then look at BMP signaling in these mice, what we see is that markers of BMP signaling, such as SMAD7, are suppressed in the high line significantly, and there is a trend towards suppression in the medium line, but it's not detectable in the low line, and that's congruent with the effect that the overexpression has on hepcidin. So there's significant suppression of hepcidin in the high line, and the SMAD7 effect is consistent with it being mediated by BMP signaling. Just as in the hemochromatosis models, where increased serum iron concentrations and increased liver iron concentrations are accompanied by an increase in hemoglobin, there's a slight but significant increase in hemoglobin noted both in the medium and the high line. And the red blood cell count is essentially unchanged, so this is mostly due to increased hemoglobin content of each individual cell. But interestingly enough, so everything I told you so far would be expected. What we did not expect is that there were multiple other somatic features in these mice. And specifically, the transgenic males in particular did not, many of them did not survive to weaning, so that we had decreased survival of the transgenic males, and I will show you the data. The males that survived had smaller body size, and even the females had smaller body size, although the effect was smaller. The Erythropoiesis overexpressing animals had smaller kidneys and impaired renal function, and smaller kidneys even when this was indexed to their body size. They had smaller gonadal fat pads, again, even relative to body size. They exhibited a partially penetrant behavioral abnormality where they were running around in circles. About 30% of them exhibited that. And they had other abnormalities that could be potentially attributed to vestibular dysfunction. And that was the abnormal writing reflex, I'll tell you about that, and abnormal negative geotaxis. So multiple other somatic abnormalities in these transgenic mice.
Tom, just a couple of questions in the chat. One is, do you know how long-lived Erythropoiesis is in plasma?
We actually don't.
Okay.
So we have not done half-life studies yet. So I don't know how long it is, how long it persists in plasma.
Okay, and the second question is from Dean Tanton, which is there a role for Erythropoiesis in regulating iron homeostasis in the developing embryo? I thought activin TGF-beta plays a role in transferrin-bound iron uptake in the placenta. It may be a phenotype only unveiled in iron-limited mothers. That's the question, kind of. Is that?
Yes, so does erythroferrone play a role in the embryo? So we have some evidence that it is important in embryonic development in the xenopus. So it was overexpressed and knocked out in the Xenopus embryo, and there were multiple vascular abnormalities seen in that. We have not seen anything like that in mice, and it is the Erythroferrone knockouts, as far as we can tell, are born completely normal. They become transiently anemic as they develop. So there is a transient anemia seen at 3 weeks, but as far as we can tell, the Erythroferrone knockout embryos and newborns are completely normal, but this is not true in Xenopus.
Okay, one more question from Laura Silvestri. Is Erythroferrone affecting erythroid proliferation and differentiation in the transgenic mice?
The short answer is we're working on it. And the longer answer, we have not seen anything obvious other than the increase in hemoglobin, which would be expected from the iron effect alone. But we are looking at this in more detail. So we will have more data on that later. But at this point, there's nothing dramatic that we observe other than the increase in hemoglobin, which could be entirely due to the iron effect.
Okay, thank you.
So here's the survival data. So these are bar graphs, and you can see this is the relative survival of the pups in line L, M, and H. And this is the survival to 3 weeks, to the weaning point. And as you can see, the low line is relatively normal. There is a loss of males, but it's equal in the transgenic and wild type. But if we look at the high line, there is a significant, statistically significant deficit in males that survive to weaning. And we don't yet know exactly why that is, but trying to figure it out. One possibility is that the vestibular dysfunction or whatever other abnormality in the balance and position sensing there is, prevents these mice from feeding normally. I mean, that still remains a possibility, but may not be the only explanation. So there's a male deficit, male transgenic deficit in these mice. And that deficit is not present if you look at embryos. So if you look at preterm embryos, you can see that the distribution is more or less as expected. So the loss of males occurs after birth, and it's specific to, as you can see, is sex specific. Here are the growth curves, and the growth curves are relatively normal. And the top line is the males, the bottom line is the females, and you can see that the red transgenic line and the open symbol, a wild type, are right on top of each other in line L. They begin to separate in line M, where the transgenic males are smaller, already in line M, and there is a statistically significant but barely visible decrease also in females, and this becomes greater in the H line. So the Erythroferrone overexpressing mice are quite a bit smaller than the wild type mice, and this is true whether or not they manifest that spinning behavior. So there's no correlation between the spin behavior and the size of the mouse. So the effect on size is separate from the effect on the behavior. And this is present in both males and females, but very pronounced in males and barely detectable, but statistically significant in females.
I'm going to interrupt one more time. If it gets too distracting, just say, let's hold on.
Okay.
Mark Fleming wants to ask, is the lethality just a transgene insertion effect? Have you done WGS to find insertion sites?
Yes, I will show you the insertion site. We'll return to that question.
Great, thank you.
So, so here is the effect on some of the internal organs. So most of the internal organs of these mice, the transgenic mice, are proportional to the animal size. But if you look at the gonadal white adipose tissue or the kidneys, there's a significant decrement in the size of both the fat depots, the gonadal fat depots, and the kidney size. The red symbols are the transgenic mice. The white open symbols are wild-type mice, males, females at 6 weeks, males, females at 16 weeks. Same for the kidneys. So both the gonadal fat pads and the kidney size are smaller, but inguinal fat pads, liver, heart, and spleen are more or less the same size. Now, is this difference in kidney size actually functionally important? And it turns out that it is. These mice, the transgenic mice, have impaired clearance of urea from the circulation. So they have increased urea concentrations in blood. And that's true for females as well as males. It's true at 6 weeks. It's true at 16 weeks. So the kidney size, the decreased kidney size is actually functionally important. So here are some of the behavioral abnormalities. So the riding reflex is if you take the mouse and you put it on its back or throw it on its back, before it lands, it will essentially turn itself upright and land on its feet. Negative geotaxis is when you point the mouse on an inclined plane downwards, it will turn around and start climbing back up. That's called the negative geotaxis reflex. And as you can see, most of the transgenic mice have abnormalities in geotaxis or in riding, and that's relatively rare in the wild-type mice. And here is the behavior of the mice. As you can see, they have this compulsive circling. Tends mostly to be to one side, but sometimes they do a figure 8 as well. So this is their usual behavior, and that affects about 35% of these mice, so it's only partially penetrant. Let's see. So to follow up on Mark's question, could this be an insertional mutagenesis effect? So we sequenced the genomes of these mice and focused on the highest expressing mice. And so I will show you where the insertion is. It is on chromosome 4. The only nearby RNA is a non-coding RNA not known to be involved in any of the phenomena that we have analyzed. And as you can see, so what you see here is short reads, 150 base reads, superimposed on the chromosomal sequence. And so in areas where you inserted the transgene, there could be missing reads. And in fact, what we see here is that the transgene replaced a part of the chromosomal DNA, and this effect is heterozygous. And there are 3 copies, so we can actually look at the junctions here, and there are 3 copies of the transgene inserted into this area, and they're inserted head to tail. And as far as we can tell, this non-coding RNA has no role in any of the phenomena that that we described here. But of course, it could have some unknown, as yet undescribed effects. But yes, that non-coding RNAs, in fact, could be disruptive in a heterozygous manner. So to summarize these studies on the earthy transgenics, earthy overexpressing mice develop iron overload as we predicted if they would. Unexpectedly, they have a number of other phenotypic abnormalities. These are not transient. They appear to persist to 16 weeks. And some of these may have counterparts in severe ineffective erythropoiesis, specifically in congenital anemias, such as pyruvate kinase deficiency or severe alpha thalassemia, where you can see stunting, renal abnormalities, neurodevelopmental delays. And there is at least a possibility that some of these abnormalities in human congenital dyserythropoietic anemias or human anemias with ineffective erythropoiesis could be also caused by very high levels of erythroferrone. And the mechanism that we propose could be mediating these developmental effects. would involve the decrease in BMP signaling during development in organs that require the BMP signaling for normal development, such as the vestibular apparatus BMP4, the kidney BMP4 and 7, the pituitary BMP4, and adipogenesis BMP2 and 4. So, so the same mechanism that we propose is driving hepcidin production in the liver is also important in the development of organs. And erythroferrone, when it is expressed during development at high level, could be affecting the organogenesis in these organs. One point that I just have to emphasize is that this model, the beta globin driven ERFE expression in the mouse initiates ERFE synthesis someplace around mid-gestation, which is different from human beta globin, which would begin to be expressed after birth and during early postnatal development. So, so again, beta thalassemia is less likely to register these abnormalities in humans, but other congenital anemias with ineffective erythropoiesis could have defects that are partially mediated by the overproduction of ERFE, and that's something that is worth investigating. So overall summary, I hope I showed you some evidence that ERFE is an erythroid regulator of iron metabolism. It is secreted by erythroblasts and acts on the liver to suppress hepcidin. It acts at least in part by binding to BMPs and decreasing BMP signaling. Overproduction of ERFE causes iron overload in the transgenic model. High-level overproduction of ERFE has adverse systemic effects on multiple targets issues, probably developmentally mostly. And some of the systemic complications of severe anemias with ineffective erythropoiesis could be caused by ERFE overproduction. And based on these observations, we would propose that targeting ERFE may be a useful therapeutic strategy. And I thank you for your attention. I have to credit a large number of people involved in these studies. The leads on the first part was, the lead was Leon Kautz, who is currently leading a research group in Toulouse, and Richard Coffey, who is completing his postdoctoral fellowship in our group in Los Angeles, was responsible for the second part of the presentation. And thank you for your attention. Happy to answer questions.
Thank you, Tom. We have a couple of questions in the chat. I can read them, or I can have people ask them. Mark Fleming, do you want to ask your question?
Sure. Hi, Tom. My question is, I mean, it sounds like this is probably going to be just a BMP ligand trap that has pleiotropic effects. Are the homozygous transgenics lethal?
We have not looked at homozygous transgenics, but we did look at CMV promoter-driven transgenics, and we could not recover any mature animals from that. The lines died out.
Okay. Jan Christian has a comment, I think. Jan, do you want to comment?
Sure. It's just we've been studying BMP4 mice for a long time, and they show the null BMP4 null heps show severe circling, not completely penetrant, severe kidney defects, and also post-embryonic lethality. But oddly, with BMP4, the lethality is completely restricted to females. And we're trying to figure out why that is and what they're dying from. Oh, and runting.
At least for which BMP?
BMP4.
4.
And so, it's also on a C57 black 6 background.
Yes.
Only?
Yes. Yeah, the sex effects here are very interesting, and the sex effects in BMP signaling are poorly understood. And yours are inverse of ours. Interesting. I don't know how we'll put that together. But thank you for your comment.
Okay, a comment from Ann Smith. Do you want to mention that, Ann?
Oh. Sorry, I was just slow there unmuting. Hey, Tom, thank you. That was a great presentation. Seeing the animals circle clockwise, I had a question and a comment. Do all of the animals that exhibit that behavior all turn in the same direction clockwise?
I don't think they're in the same direction, and only 30% or 35% exhibit that behavior of the high expressors.
Okay, so I just remembered, sadly in a way, I had a dog with a brain tumor. And when we took the dog to the neuro dog specialist, they said that if the tumor was on one side of the brain, then the animals would circle in clockwise and the other side anti-clockwise. And I just wondered if there was any kind of links there, any relationships that you had either discovered or thought about, or if you knew of that. So
we don't think there's a preference to sidedness. I think in most animals mostly circle to one side, but sometimes they do figure eights. And there doesn't seem to be a single preference, and we have the brains and we're studying them, so we hope that we'll be able to figure out what's going on here.
Yeah, thank you.
Okay, a question from Melanie Castro. Melanie, do you want to ask your question?
Sure. Hello, Dr. Ganz. It's Melanie. Thank you for your presentation. I just wanted to ask you if the small phenotype of the transgenic implies that the bone mineral density from these mice were lower as well, or you don't know yet that question?
So I specifically avoided talking about bones because we're still studying them. What we very early saw, and I would like to wait for the complete study, but what we saw very early is that there may be a defective bone mineralization in the high expressing animals. So there is more bone that's not mineralized in the high expressing animals.
Thank you. A question from Clara. Hi, Clara.
Hi. Tom, I'm puzzled by your idea of ineffective erythropoiesis associated anemia with very high Rph and that is responsible for some of the abnormality you have described. You show an impressive level of Rph in non-transfused thalassemia. Have you measured Rph in other types of anemia with ineffective erythropoiesis? And yes. Can you say that there are other types with the Rph so high as in non-transfused thalassemia?
Yes, and in fact, some of the highest levels we measured were in a patient with pyruvate kinase deficiency. So it seems to be not just thalassemia, but other anemia, other ineffective anemias with ineffective erythropoiesis as well. We don't have a lot of data, and we would be interested, of course, in your country has the best collection of patients with anemias with ineffective erythropoiesis in the world. So by all means, we should measure patients with CDAs. We haven't done that.
Thanks.
Great. Thank you. I don't see any more comments in the chat. If I have missed any, please go ahead and unmute yourself and ask them. Yeah, I think that's it. So I'm going to just thank Tom again for the wonderful presentation, and thanks for everyone's participation. I'm going to share my screen to remind you that the next talk in this seminar series will be given by James Cox. He is the director of the Metabolomics Core at the University of Utah and is going to speak on challenges and solutions in erythrocyte metabolomics. And if you haven't hopped on the metabolomics train, I highly recommend you listen to James's talk because it's pretty interesting. We're learning a lot about it, and I hope to invite you there. For those who are participating, the password and the Zoom invite is the same. And so we look forward to hearing from all of you. I'm going to share one more screen to just remind you again about the, let's see if it does it. Share screen, there it is. Okay, to remind you about applying for these potential fundings for those of you who are able to do so. The funding opportunities that are being offered through the NIDDK CCEH, and you can find that imaged here on the website at cceh.io.
Okay. Okay. Yeah. Okay.
Okay, and thank you, everyone. Thank you for the talk, Tom, and hope everybody is well. And that will, with that, stop the presentation. Thanks again to everybody, and we'll
See you next time.
talk to everyone later.
Okay.
Thank you.
Okay, bye-bye.
Thanks,
Bye.
Tom.
Thank you very much.
Okay. So coming back to
Thanks. Yes, I don't think yours are around.
yeah.